Hi, I'm Lena Trogdon, and my protein is acetylcholinesterase, and here is my model. As shown in the video, I am forming blue Play-Doh into tiny little micromolecules and forming them together to make the shape of the molecule as a whole. And I also use red Play-Doh to form the active site. This is the model, acetylcholinesterase. As you can see, it is surrounded with blue structure. This is the molecule itself. But if you look in the middle, there's a small red section. This small red section is the venom in the active site. If you look at the model, you can see I created it into an irregular shape, including the tiny micromolecules. I formed the model to be similar to this photograph of acetylcholinesterase. I included the curve on the end and the rounded structure of the molecule. To talk about the questions that are involved with my model and project, the first one would be, list your assigned protein and mutation. So, the assigned protein is acetylcholinesterase, and this is not a mutation, but it is a chemical change which occurs when serotoxin, which is snake venom, binds to the protein of an active site. The second question, what is the normal function of this protein? Categorize the type of function that this protein holds, which is structural, enzyme, transcription factor, receptor, relay molecule, and etc., and explain why you chose this. This enzyme's sole responsibility is to break down the neurotransmitter acetylcholine. The substance involved is produced by a living organism which acts as a catalyst to bring about a specific biochemical reaction. Number 3, where is this protein found? Include the subcellular location and the type of cells that typically contain this protein. The major form of acetylcholinesterase is found in the brain, muscle, and other tissues. The organism that this is found in is electric fish, and acetylcholinesterase is found in the synapse between the nerve cells and the muscle cells. Number 4, categorize the type of change the mutation has on the protein function and explain why you chose this. So I chose this option because the mutation or chemical change in this protein only occurs when the toxin binds to the protein's active site. And from a quote in the source that was provided for us, it says, more recently, however, it is thought that hydrophobic interactions are equally, if not more important, in binding this region of the substrate to the enzyme. And then moving on to sentences down, what remains is an active form of the enzyme ready to react with newly released molecule of ACH. So I used that quote to just back up what I had just previously stated. And question 5 was to create the model, which we have seen. Question 6 would be, what are the main functional groups present in the AA side chain? And I put acetate and choline. And then, is it hydrophobic or hydrophilic? And ACHE, which is acetylcholinesterase, is both hydrophobic and hydrophilic. It exists in several forms. While others can be hydrophobic, the other can be hydrophilic. Is it neutral, positive, or negatively charged? It was thought to be negatively charged in the past, but now recently it's thought to be neutral. And how would you characterize its size compared to the other 19 strands? I would characterize its size as small compared to the 19 standard AAs. And the next questions is, what are the main functional groups present? That would also be acetate and choline. And mutated AA side chain is hydrophobic and positively charged. I would characterize it as large compared to the other 19 strands. Discuss the differences in the types of reactions that mutated AA side chain. So now it's talking about the differences between the original chain and the mutated chain. So I put, while the original chain is neutral, the mutated chain is positively charged, binding to the active site. Another comparison would be that the fact of in the original and mutated, one is smaller and one is larger than the other. How does your assigned mutation affect the protein function, the function of the cell, and the function of the organism? The assigned mutation has poisons and toxins that attack the enzyme. It causes acetylcholine to accumulate in the nerve synapse, and it paralyzes the muscle. So the limitations of my model will be the fact that it is not a mutation like the other 4 groups. It is a chemical change. So that makes it different whenever I'm trying to find information on changes or mutations. Overall, this was an interesting project to do, to understand the effects of snake venom and how it can affect the body or the body of an electric fish too. Thank you for listening, and that is the end of the video.